All right, good afternoon. I am Dean Jessica Berg, and I'd like to welcome you to our Schroeder Lecture, our premier lecture series for our Law Medicine Center. We have a fabulous speaker today, and to introduce her is Professor Sharona Hoffman, who's the co-director of our Law Medicine Center. So I'll hand it over.
Yes, please turn off your cell phones, etc. Well, welcome. It's my pleasure to introduce Dr. Christine Grady, who is a nurse bioethicist and a senior investigator at the National Institutes of Health and also the chief of the NIH Department of Bioethics. In her research, Dr. Grady focuses primarily on the ethics of clinical research, including informed consent, vulnerability, study design, recruitment, and international research ethics, as well as ethical issues faced by nurses and other clinicians. Dr. Grady has authored more than 175 papers in the biomedical and bioethics literature and authored or edited several books, including the Oxford Textbook of Clinical Research Ethics. She served from 2010 to 2017 as a commissioner on the President's Commission for the Study of Bioethical Issues, and that is the US President. Her work is known internationally, and she has lectured widely on ethical issues in clinical research and clinical care. She's an elected fellow of the Hastings Center and of the American Academy of Nursing, a senior research fellow at the Kennedy Institute of Ethics, and an elected member of the National Academy of Medicine. She has participated in numerous intergovernmental task forces and is the recipient of several important awards from the NIH and elsewhere. She even has a really famous husband. She is married to Dr. Anthony Fauci. I got to know Dr. Grady last year when I spent a sabbatical semester with the NIH Bioethics Department, and it was an absolute delight to work with her. It is a great pleasure to welcome Dr. Grady to our law school, and we look forward to her talk, Limits of Biomedical Research, Whether, Why, and How.
Thank you so much for that nice introduction. It's nice to see so many faces here, and I'm glad that the dean said my talk is going to be fabulous before it even starts. So, I'm never optimistic. Hopefully I'm not going to disappoint. But I hope that you will join me on sort of an inquiry, an intellectual inquiry about this topic of limits on biomedical research. That's what I'm going to talk about, and these are my views, not the NIH's views. Let me see if I can. Oops, did I do the wrong thing? How do I go back? Okay, there we go. Okay, I want to start by recognizing something that probably everybody knows. Biomedical research aims to advance our understanding of health and disease and to identify ways to prevent or treat or ameliorate diseases. This is how we make progress in life, right? The findings of research have had and have the great potential to have a real impact on our lives. And we have a few examples, recent examples. Treatment for HIV as one. Treatment for childhood leukemia is another example. Statins might be a good example. COVID vaccines might be a good example. So there's a number of things, and we could talk about that. But arrow key, I tried that and it erased everything.
I think you're okay right now.
Okay.
The right arrow key.
Right arrow key, thank you. Thank you. Okay, but the question that I want to explore with you tonight is this one. Is all biomedical research that promises progress in understanding health and illness justified? Or should there be certain areas of study or certain topics that are off limits? So Dan Callahan, who was the founder of the Hastings Center, wrote a paper which was only published posthumously, where he talked about progress. And of course, biomedical research is progress, right? Progress has given us most of the ethical and social benefits of modern medicine alongside the most aggravating ethical challenges. And so part of what bioethics is about is trying to find the best balance of these benefits and harms of progress. So I want to ask the question, should there be limits? And if so, should they be hard limits like a red light? Should they be a yellow light? Should they be a green light? And I think we're going to find that there are people in the literature who have called for all three of those. One is David Baltimore, who's a biologist, Nobel Prize winner, was the president of Caltech for a number of years, is now emeritus. And he wrote a paper in 2005, I think he had a prior one that was very similar actually, and asking the question, should limits be placed on biological research because the outcome might be detrimental to the established or desired societal norm or order, or the relationships that characterize our contemporary society? And he went on to argue in this paper, there should not be limits, and there cannot be limits, that they're impractical, they're never feasible, and they're really disruptive to the whole order. And he had a number of what he called risks that limiting science would pose. The first one is one he called error of futurism, which is basically the idea that we don't know enough about the future. We cannot really understand what's going to happen in the future. And so any decision now about what's wrong that might impact the future could be based on misunderstanding of what's really going to happen. A second one, he said, is that biomedical research is like a renewal process, and this limiting it disrupts that process in a way that really could be detrimental. It might be detrimental not only to our understanding things, but might be detrimental to the extent that biomedical research is a part of a renewal process that maintains the fascination of life. A third one, and there's a lot of words on this slide, but the third one basically was limiting biomedical research could lead to fear, mistrust, and unrest, because a lot of the limits are going to be politically motivated or ideologically motivated, and pushed by certain people, and therefore could cause distrust in the whole system. And then, of course, he said, it's impractical. It's impossible to limit research. In fact, what he said in the end, I think I might have another slide. No, I don't. What he said in the end is we could do more or less research, but we can't choose the types of research that we do, is what his argument was. Now, this is a quote from a report of the Presidential Commission that Sharona mentioned that I was part of. Was written in 2010 about synthetic biology, and we put a number of sort of guiding principles for emerging technology in place. One of them was this one, called intellectual freedom and responsibility. And the commission argued that democracies depend on intellectual freedom, coupled with the responsibility of individuals and institutions to use their creative potential in morally responsible ways. And went on to talk about how important it was for progress that we leave people free to pursue their scientific interests, especially understanding, historically that a lot of scientific discovery, a lot of progress that has been made really was the result of serendipity or individual scientists, pursuing their interests. I want to point you, though, to the last sentence of the second paragraph, because what the commission did say was, at the same time, science should reject the technological imperative, meaning the mere fact that something can be done does not mean that it should be done or ought to be done. John Harris, who is a philosopher at Oxford, has written in several times, but in this paper he talked about research as an ethical imperative. And he said, biomedical research is so important that there's a positive moral obligation to pursue it and to participate in it. He went on to say, we can't neglect it, we've got to do it, but he put some limits on it. And you can see, again, the last sentence of the second paragraph. He says, research is both permissible and mandatory where the importance of the objective is great and the risks to and possibility of exploitation of fully informed consenting subjects is small. So those are the kinds of limits that he put on in his We also know, I mean, many of you know the work of Hans Jonas 50 years ago, who talked about biomedical research as an optional goal, that progress is optional, that, you can read his words here. It says, a slower progress in the conquest of disease would not threaten society, grievous as it might be to those who have to deplore their particular disease that isn't yet conquered. But society would indeed be threatened by the erosion of those moral values whose loss, possibly caused by too ruthless a pursuit of scientific progress, would make its dazzling triumphs not worth having. So he went on to say, not only is progress through medical research optional, but even the noblest purposes, not even the noblest purposes, abrogate the obligations that we have to people that we engage in biomedical research and to society. So those obligations are really what has been the focus of research ethics for many years now. And I'm going to say what they are. What are those obligations? Well, the obligations include promoting responsible and useful research that has scientific and social value, protecting participants in that research from unnecessary risk or burden, and respecting the rights of the individuals and protecting them from the possibility of exploitation. And I just put a couple of references at the bottom, but basically most of research ethics focuses on these kinds of obligations that we have to individuals in studies and to society. And that's what the basis of really the ethics of clinical research is, making sure that we focus on promoting useful research while protecting the individuals that are in it. So what does that do? That actually gives us at least a few reasons to limit biomedical research. The first one being research is not important enough, or that isn't important enough to justify any of the burdens that we're asking people to take. So a term that I've loved for many years in research ethics is social value, that research has to have social value. And that came from a paper that a philosopher, Benjamin Friedman, wrote in the 1980s, where he talked about running into a colleague. I think it was a dream that he had, but it was running into a colleague who excitedly said to him, I just successfully transplanted an appendix. And in the article, Benji says to his colleague, was that ethical? And his colleague said, well, I got the guy's informed consent. And the whole point of it was, unless there's something valuable to learn or do by transplanting an appendix, that's not an ethical study, even if the person agreed to it. We also talk about sort of Me Too studies that are done where we actually already know the answer to the question. Those don't have very much social value. And then sometimes we have sort of the debate between what's the social value of a particular study compared to the risks that it poses. Some of you may have followed during the early days of COVID, or actually all through COVID, a lot of debate about challenge studies, human challenge studies. The argument was that if you did challenge studies basically infecting people with COVID and then testing different interventions, if you did a challenge study, you would increase the social value because you'd get answers to those questions much more quickly, and you could be able to develop vaccines much more quickly. But the question was, how much more quickly would it really make a difference, and what is the risk to the people that you would be infecting, especially at a time when we didn't know very much about how people got infected, how sick they could get, and we had no interventions to treat them. So another reason that we do limit clinical research or biomedical research is that poses unnecessary or excessive harms to participants. And I think we could say this is both human participants and animal participants. And there are a lot of historical examples that are useful to think about, and the Nazi experiments as one, the Guatemala STD studies as another, but lots of examples where the amount of harm that was imposed on individuals in the studies was so egregiously great without the social value equivalent that those would not be done today. And then, of course, there are studies that would violate participants' rights, exploit them, or inappropriately harm them. And so this could be a set of studies that you might imagine where somebody is enrolled without knowing that they're being enrolled, without their consent, or they're coerced in some way or compelled to participate. Or maybe some people used to use the example of enrolling homeless people from a shelter into phase 1 drug development studies, exploiting them in that way. I think the interesting thing is we do limit research in all of these ways, and yet we still have lots of debates about what is social value, what is enough social value, how much risk is this really, and a number of other, and what does violate principles. I mean, we do allow research, some research without consent, for example. And so, you know, the balance here is not always so clear, but these are reasons that we accept. So what about the rest, though? What about not the social value of the knowledge or the risks to the individuals, but knowledge that might impact humanity or might impact the way we think about humans or pose threats to our moral values or things like that? Those are the kinds of questions that are harder to deal with. And so this set of authors, Marchant and Pope, wrote in more than 10 years ago now, that the kinds of limits that I just described are really limits that are sort of what they called incidental regulations that limit when and how scientists do research, but not on the substantive questions of what research is trying to learn. And others, this is Leon Cass in the same issue of Journal of Science, Engineering, Ethics. He noted that, you know, the sort of growing opportunities to manipulate our bodies, our minds, not merely to heal disease, but to other things, enhance people or satisfy desires or control deviant behavior. These make urgent questions about how we should regulate, how or whether we should regulate their use, not merely to assure safety, but also to, as he says, safeguard our humanity. So what I'd like to do, so the question is about these kinds of things. You know, are there kinds of knowledge that we should not pursue in the context of research because they harm our communities or our society, or they disrupt our societal norms or values in some way, or they have an impact on human dignity, or there's likely to be or going to be significant public backlash that would interfere with trust in research? And so we might think of some examples here. One example that I think is interesting to think about is, you know, the harming communities. I mean, there's some, again, historic examples that we could use, like the Havasupai tribe in Arizona, who were aggrieved because there was research trying to understand their origin stories. You know, the studies of diabetes and other things that they were doing with their samples was okay, but trying to understand their origins was not okay. There's, again, there have been historical examples of studies trying to look at the association of race and violence, and those were, you know, thought to be not appropriate. There are studies that look at identifying what some people would call deviant behaviors, and so the question, are those okay? Are those legitimate? What about disruption of social norms? What about trying to keep people alive well past 100 years old? Is that a good thing? Is that something we should be doing? What about creating chimeras, human-animal combinations that sort of change the way we think about who's human? What about germline editing, which affects next generation people? Human dignity. Some people say creating embryos is an affront on human dignity for research purposes. Some people think brain transplants. I mean, there's this whole movement of people who want to take a brain from one person and put it on the body of another person, and there's something about that that gets people to go, ooh, you know, what do we think about that? Is that something we should be doing? And when you think about public backlash, I think about, for example, primate research, something we've been doing for a long time, but increasingly it is less acceptable to the public. Now I'm going to dig into a couple of examples. They're selected examples, and I'm going to dig into them just a little bit. I can't say everything about all of them. The first one I want to talk about is heritable genome editing. And I want to, oh no, sorry. I put in a synthetic biology slide first. Sorry, I forgot I had it there. Synthetic biology I want to talk about first. This was something that we dealt with at the commission that I mentioned already before. And it was brought to us because Craig Venter, who was one of the people who mapped the human genome, had created a bacterial cell synthetically. So he created a live cell synthetically. And the response to that was, as you can see, did he create life in a test tube from a computer? And it raised the whole question of synthetic biology. And a lot of it was concerns about playing God. Can we really create life? But there are also lots of concerns in synthetic biology about risks to the environment or risks to the next generation. At the same time, synthetic biology has the potential to do a lot of really good things. One of the examples that I remember the best that I loved was artemisinin is a medication that treats malaria, which is a very bad disease that affects many people around the world. And it's hard to make because it's made from a plant. And so synthetic biology made the possibility of manufacturing large amounts of artemisinin in yeast cells, because yeast apparently proliferate very rapidly. And that experiment was being talked about at this time in 2010. It was done successfully. Sanofi has this way of making this artemisinin, and I just read the other day, it's not being used. And why is it not being used? Market forces. So that's an example of how we can't predict always the future. All right, now I'm going to talk about heritable genome editing. And I want to say, this is an example, again, of the use of a technology that's really very valuable. Gene editing and genetic engineering has been going on a long time, but as you all know, scientists discovered CRISPR-Cas9 and got a Nobel Prize for it in 2020, right? And it's an incredible tool for editing genes and can be used and is being tested in research right now to cure diseases like sickle cell disease. Now, we don't know if it will, but that's the kind of research that's being done. But at the same time, it can be used to edit embryos. And this gentleman, I don't pronounce his name correctly, so forgive me. I think it's He Jinkyu, a Chinese scientist who kind of shocked the world in 2018 because he said he had edited 2 embryos with CRISPR-Cas9 to prevent those 2 babies from being susceptible to HIV. He deleted or intended to delete the CCR5 gene. And so he announced it to the world, and the world went crazy because it was like, this is premature. We don't know enough about germline editing or heritable genome editing. And there's been lots of really interesting follow-up to what happened to those 2 babies. Some of them saying they've benefited in certain ways, others that they've been harmed in a number of different ways, in addition to the off-target effects that people worry about with CRISPR. I would call it CRISPR. CRISPR. One of the things that I find is so interesting about CRISPR is, and people say, is one of the things that's great about it is it's so easy to use. You can buy a CRISPR kit on Amazon. It's less than $200. So if you know enough to be able to use it, you can buy a kit and do your own gene editing in your garage. So what was the response to Hay's work? A huge outcry, actually, among the scientific community that said, and some of the names on here, some of the authors on here are the people who won the Nobel Prize, or the people who basically discovered CRISPR-Cas9. And they unanimously actually called for a moratorium on heritable genome editing. So not not somatic gene editing, not the sickle cell example, but anything that might be passed on to future generations. And one of the things I wanted to show you was this is a statement from the NIH director, which actually predated that experiment that I just talked about. The red parts are things that I made red. Basically, this is a statement that NIH will not fund any gene editing in human embryos. But there were several reasons given, and that's what I want to point to in the red. One of them was, it's a line that should not be crossed, without any further explanation of what that means. But that's kind of the thing about, affecting humanity or our values or whatever that I was talking about. The other reasons given are very consistent with the ones that I started with. Risks, serious and unquantifiable risks or safety issues. Ethical issues by altering the germline in a way that affects the next generation without their consent, so violating their rights. And the third is a current lack of compelling medical applications, meaning we don't know enough about the social value. So those reasons are also relevant for limiting heritable gene editing. Interestingly, there was a paper not too long ago, I think it was 2020, it was Francoise Baylis and others who looked at laws and guidelines in countries around the world. They looked at 96 countries, and you could see that almost all of them prohibit heritable genome editing. So there are rules and guidelines and laws in a number of different places. Another example, organoids and embryoids. So organoids, maybe you all know about these. These are really exciting progress, scientific progress in current discussions around scientific communities. These are 3D structures that are like mini organs. So they're made to resemble human organs. And they're made from pluripotent stem cells. And they can be used eventually for three major things. One is for fundamental research. So when you really need to understand an organ and you can't get inside somebody's body to cut it up, you can have an organoid of that organ and learn what the organ does better and how to both physiology and pathophysiology of that organ. So that's one. The second is translational research, testing interventions in organoids. And in fact, some people believe that eventually we will have the need for less animal research because we'll have organoids to test drugs and things in. And then the third major category of use will be regenerative medicine, that we could perhaps imagine an organoid that can be given to a human and replace a damaged organ that they have. But where the action is in terms of the question of limits is in neural organoids. And neural organoids, neural transplants, and neural chimeras. And why is this? Well, why do people care about this more than like a kidney organoid? Because there's a way in which the brain is, sort of the essence of who we are. And so people kind of worry about this combination of things going on. So this was a study that was finished, just about a year ago at the end of 21 by the National Academies, where they looked at this emerging field of human neural organoids, transplants, and chimeras, and put together, I think, a really thoughtful report that basically says they found a number of things. There's a lot of value to this kind of research for all the reasons that I just gave you. You could learn a lot about the brain and the nervous system that is very hard to learn in other ways. They noted that the state of science is still pretty early, but moving fast, evolving very quickly, that there are a number of issues of ethical concern, which I'll get at on the next slide. There's a concern about consciousness. If you have a neural organoid, how do you know whether it could be conscious? And we don't have good ways, we don't understand really consciousness that well, and we certainly don't have good ways to measure it. There are questions about oversight and regulation. These are not humans, they're not under human subject regulations. They're not animals. They're not under animal regulations. They're a little bit out of the loop in terms of who's paying attention. And they said there's a great need for public engagement and communication, because this is something that's happening and going to happen more quickly over the next years. So a couple of things about their list of ethical concerns. I think they're really useful to look at. So the first is, brain disorders in general are really bad and hard to study. And so the limitations on our ability to study them in humans themselves or in animal models is a very strong reason that developing organoids and even chimeras might be a way to solve that problem. But there are moral, ethical, religious concerns about mixing humans with non-human animals. Hubris, affront to dignity, blurring the lines between what is a human and what isn't a human. Other issues, they say, some people might object to the use of their specimens for neural organoids. And a lot of times the consent to give a cell to make an induced pluripotent stem cell is not necessarily described in the context of what are we going to do with that induced pluripotent stem cell. It's not always even known at the time that the cell is taken from a human. So humans are not going to know. They also said it could be concerned that we might make a lot of very distressed animals if we give them more human-like characteristics. And then the issue of consciousness. It's possible that these groups of neural cells, as they get stronger and bigger and more evolved, could develop awareness or sentience or consciousness. Everybody's worried about that, but again, how we would know is the interesting question. So there was, this is a small study, but it's an interesting one. They interviewed 28 people and asked them about how they would feel about using their cells for developing cerebral organoids. And you can see, this is what they were worried about: the possibility of consciousness, the possibility that some of their own personal characteristics might be revealable eventually, and also potential misuse. And they said they would support it under conditions of adequately informed consent and some pretty significant oversight. And as I mentioned in the last slide, we don't have either of those really for this area yet. Another thing is the report, as you saw, was about not only organoids, but transplants and chimeras. And I think there's some really interesting questions about these two things, especially in the neural space. There have been some studies, for example, that have been able to take human brain cells, and implant them in the brains of mice and rats. And in a very recent study, it was just like a couple of weeks ago, the human cells not only were assimilated into the rat brain, but grew. And so over a very short period of time, about a third of the rat brain was now human brain. And as somebody said, I think it was Altacharo said, there's something really discomforting about, a human brain like stuck in the body of an animal, or an animal stuck with this part of their brain being human, not really knowing what to think about it or how to deal with it. And of course, we can't ask the animals, but it made me think of, and this is like to give you something fun to think about, a book that I read, or my kids read years ago, and I love, called Mrs. Frisbee and the Rats of Nim. How many of you have ever read that book? I would recommend it. It's a great book. And I took out one quote from that book, which is example of this kind of animal distress that the National Academy was worried about. And this, the stories about rats that got smarter, basically. So this rat says, "We don't know where to go because we don't know what we are. Do you want us to go back to living in the sewer pipe and eating other people's garbage? Because that's what rats do. But the fact is, we're not rats anymore. We're something Dr. Schultz has made, something new, something." He says our intelligence has increased more than 1,000%. I suspect he's underestimated We're probably as intelligent as he is, maybe more. We can read, and with little practice, we'll be able to write. I mean, to do both. I think we can learn to do anything we want. But where do we do it? Where does a group of civilized rats fits in? So this is a great, I love this sort of story because it kind of pushes your thinking about this. Okay, these are rats. Some people would say, who cares about rats? But we're doing it to higher level animals as well. All right, another puzzle, dual use research. Now, this is a document from the World Health Organization which basically defines what dual use research is. Basically, any research that can be used for nefarious purposes, right? And they point out, and I think very importantly point out, most of the focus of dual use research has been on the life sciences, on microbes and developing microbes. But there's no reason it has to be limited there. We can imagine lots of other kinds of research results that could be used for nefarious purposes. There was, again, a National Academy report, this was from 2017, that talked about dual-use research of concern, which is a very specific kind of dual-use research, focusing on a certain set of pathogens, and they name the pathogens in this report. And basically, they say in the middle of this paragraph, these policies build on the fundamental idea that basic research should be open, so sort of green-lighted, but should be subject to policies that provide for the assessment and mitigation of risks in certain cases. So there's the sort of, how do we decide which ones is my question. I found this paper, and I think it was really fascinating. This is a paper by a couple of modelists who are developing artificial intelligence. And they wrote this paper, and the first sentence of the paper says, "The thought had never previously struck us. We were vaguely aware of security concerns around work with pathogens or toxic chemicals, but that didn't relate to us. We primarily work in a virtual setting. They work with computers." But what they had done was they had developed a molecule generator guided by machine learning to be able to find, process through lots of different compounds to sort of select for those that had the least toxicity and the most targeted activity. And so then they did this experiment where they just flipped the logic around. And they were able, he said, in 6 hours to generate 40,000 toxic chemicals that were as toxic as some of the really dangerous nerve agents. And started to, raise the issue of people who are doing machine learning and AI, they don't think they're subject to a dual-use concerns, but his example was maybe they are, and maybe we should be thinking about that. That's just another part of their story. The President's Commission, as I mentioned, talked about synthetic biology, but one of the things in this intellectual freedom responsibility principle that they mentioned was that general concerns about the potential malevolent use are usually not sufficient to stop certain kinds of research. And if we stifle the kind of research that could be eventually used, we might be at a loss. We might be hurt because we won't understand it well enough and won't be able to combat it. So, I won't read the words, but that's what that says. So, what do we do about this? And going back again to this Leon Cass paper, I think he asked some really interesting questions. Would it be good or not good to prevent the discovery A, or forbid scientific practice B, or use C of technology D? Is it even possible to do so? And would it be wise to do so? So should we do it? Is it possible to do it? Would it be wise to do it? And I think the answer is, I'm not sure. I think that the idea that there should be absolutely no limits is probably wrong. That there should be some limits, maybe even prohibiting studies under certain circumstances, maybe not wholesale categories of research, but studies. And then the question is how? And, you know, limiting research, the doing of research is different than limiting the dissemination of research, is different than limiting how we apply research or how we use research findings. And so those are really important distinctions to keep in mind. So some of the safeguards and guardrails that I think are relevant to at least think about include these. And I'm going to say, you're going to see why in a second. I'm going to say the least about regulations and laws because I know who I'm talking to today. And I thought, for example, some of the literature that talks about applying laws and regulations to determining topics of research talks about some of the things actually that David Baltimore wrote in that early paper. You know, laws are blunt instruments. Science is moving fast and evolving fast. It's hard to put a law together now that isn't outdated by the time the scientific progress is made. It's also very difficult to predict the impact of certain kinds of science, as we've just heard about. It's also probably unenforceable because, you know, a law that might apply to the United States doesn't apply to the rest of the world, and all of this science is being done globally. Even the laws in the United States that protect, or regulations that protect human subjects and research only apply to some human subjects research, and so, you know, how do we do it? But through laws or regulations. And then there's the risk, other people raised the risk that David Baltimore also did of, you know, ideological pressures to do certain things and not other things. So I'm going to talk about the other things briefly. I know we're running out of time. Principles and guidance. So there were these principles that the commission put together that I think are actually quite useful. And again, it was related to synthetic biology, but it could apply to any emerging technology. And if you apply them, it might put some brakes on the research that might be done. So one of them is public beneficence, that research has to be done to maximize the public good and minimize public harm. That's not in our current regulations around research. Responsible stewardship is the idea that we're responsible not only for people now, but future people, and future generations, and our environment in which we all and our future generations are going to live. And so paying attention to safety and long-term effects of certain kinds of research makes sense to help future generations. Intellectual freedom and responsibility I've already mentioned several times, so I'm not going to say anything else about that except for one thing, and that is, in that discussion there was also this idea that we should have regulatory parsimony. In other words, use regulations where they really work and where they really help, but don't just make regulations otherwise. Democratic deliberation was a very important part of the commission. It was basically the idea that we should be engaging the public a lot more, not only in all of these kinds of research, but probably all kinds of science. And democratic deliberation is a very specific methodology, but I think public engagement is something that we should think about more broadly. And then justice and fairness, paying attention to especially how the benefits and burdens of this kind of research will be distributed. Another possibility is something that I call special scrutiny, and this was a project that I was part of more than 10 years ago now, I think, where we recognized a couple of things. One is that there are lots of research projects being done, and there is a lot of research that has social implications and long-term implications, and yet we don't have a mechanism for thinking about those. IRBs are not supposed to, and probably don't have the expertise anyway. And so there's this big void, right? Who's supposed to do it? Now, there are examples of things that have been put in place over time that I think are helpful. Advisory committees like the RAC, President's Commissions or National Commissions, National Academy's reports, I think, have been really helpful. But at least at this point in this paper, we said there needs to be a body that exists, that nationally anyway, thinks about these things, thinks about where the limits ought to be, and that it should be independent and not politically or religiously or ideologically based. So that's another example of something we should do. The third one, I think, or fourth one, I guess, is public engagement. And I really do think this is important. And again, I'm going back to something that Leon Cass wrote, where he said, oversight of these questions in our society belongs in principle to the democratic polity at large. Not only because they're affected by these technologies, but because the decisive issues for debate are matters of morals and politics, not of science or technical expertise. I mentioned what the commission said about democratic deliberation, but just let me give you some more words of theirs because I think they're interesting. It's an approach to collaborative decision-making that embraces respectful debating of opposing views and active participation by citizens. And I think that's a really important part of this, and yet in our world, increasingly more difficult to accomplish. And yet, if the public doesn't understand or doesn't have some say in the kind of research that gets done, then the backlash is going to be even greater if they find it unacceptable. And so asking the public, I picked two cartoons, but asking the public to engage, not just to listen, but to engage in discussion and deliberate a little bit about what kind of research should be done, what they think, what they value. And also to do more, we need to do more to enhance scientific literacy. I found this chart. I don't know if you can see it very well. It's kind of small. Basically, I think probably most of you know, scientific literacy in the United States is pretty low. People do not understand science. And this was not a chart about literacy. This was a chart about engagement, and engagement in something that was sort of scientific. And you can see the numbers are very small, with the largest one being helping a child with a science fair project, which, you know, a lot of people do, even if they don't understand science. But I think a lot of this includes thinking about the future of science and interaction of science and society. And they overlap with science literacy. So I'm going to end with perhaps not a very satisfying summary. I think, based on all that we talked about, I don't think there can be a red light, a wholesale prohibition of probably any category of research as wrong. But we do need to really pay attention. So there can't be a green light either. There can't be a green light. We know it would be foolhardy, really, for us to say, just go ahead, do any kind of research you want without paying attention to the potential impact on our lives, on our safety, on our lives, and on the future. So I do think the answer is the yellow light, the caution. And I think it's necessary to find ways to proceed that promises to relieve some of the, the ills of our human condition without, destroying our humanity, without really impacting our humanity in really negative ways, and to proceed with caution as we do that. And I think the ways we do that are the kind of things that I just tried to say. I think regulations and laws do have a role. They might have a small role, but they have a role. I think principles, guiding principles, do have a role. I think we need a procedure, some kind of a mechanism for thinking about these at a high level, reviewing research as it's unfolding and making decisions. And, we have National Academy reports, I referred to several, but maybe we need something else. Maybe we need something more permanent. And we need to engage the public. We need to communicate with and engage the public, prioritizing science education and science understanding and science and engagement, recognizing the many challenges that poses. And I think we need to do all of that with our eyes wide open about the kinds of good things that can come from science and the kinds of harms that could potentially come, and I mean harms in the big H way, and a strong dose of humility, because we can't see the future. We can't appreciate everything that might happen, but we can make judgments based on, wisdom that people have, and we should be willing to amend those judgments as we move forward in life and science. So we need to be wise old owls. And that's it.
So we have about 10 minutes for questions, so please raise your hands and Dr. Grady will call on you.
Try to answer your questions. Andy.
Should we use the microphone?
Yeah, oh, and please wait for the microphone because we're live streaming it.
Okay.
Very nice talk. You convinced me that there's a lot of scientific research that we ought to be worried about. I wonder if there are enough bioethicists in the world to evaluate that research and to make recommendations.
So, well, ask a bioethicist if there are enough bioethicists. The answer is never. But I don't think it's only the domain of bioethics. I think it is, as I tried to make the case at the end there, I think this is something that society at large should be discussing. And I hope that I didn't make you all think, oh, these are things to really worry about. I mean, I think there's great value from all of those kinds of research as well. It's just trying to think about how do we allow the value of scientific progress without really doing something harmful to ourselves and our future. So, but I don't think bioethicists are the only answer. I think bioethicists can help because people do think about some of this stuff and maybe can help lay out the issues and guide the conversations. But they're not the arbiters. That's my view anyway. Paul had his hand up.
Again, a fantastic talk. You answered many of the questions I had as you went through. But the thing that really struck me was the AI and the CRISPR examples, and the idea of the citizen scientist that now it's so cheap, democratizing of knowledge. A lot of our regulation, it seems to me, and I know regulation is one you didn't focus too much on, it hinges on the source of the funding or on people having accepted federal funds or people wanting to go to the FDA to get it approved. But if I'm a citizen scientist who doesn't want to market something and wants to study very dangerous viral things through CRISPR, what's the lever to keep me in line if I don't care what the bigger scientific community thinks? How do we balance keeping regulation and law have some teeth in it without being outdated or being draconian? What are your thoughts on how to?
I think that is really the challenge here, Paul, because you're absolutely right. DIY research of all kinds is very easy. AI is an example. I love that example because I mean AI is everywhere, and nobody's sort of controlling it. And I think that the answer is sort of like the answer about bioethicists. We can't be putting laws and regulations and telling people you can't do this, you can't do that, because people are going to do what they're going to do. I think we need more of a grassroots kind of response, and that's why I say, science education, discussions, public dialogue, democratic deliberation. I think, there are examples of that working and really helping people reflect on what they think and what their values are and how these things matter. Now, that doesn't mean there aren't going to be bad actors. There are always going to be some. But I think that and this is the royal we, not just bioethicists, we have to think about this. Science is a social enterprise, right? It's a social enterprise that helps all of us. And the kinds of speed bumps that we should put in the road should be things that we think are important. Susanna, did you have your hand up? Sorry, I saw those hands first.
So also, thank you for a great talk. A lot of things to spark to mind. So one of the things you implied, but I actually, in your response to Paul, I think said it very specifically, is that you believe that deliberative democracy or other approaches may fend off or reduce public outcry, right? But I don't know if that's, maybe I'm being cynical, but I actually think it might be the opposite. Yeah. And so, I know Norm Daniels and Sabin, in their book Setting Limits Fairly, like they've laid out and they've used this approach effectively. but in very specific areas. And so I'm curious what your thoughts are, if you, or perhaps you have empirical studies that I am not familiar with.
Yeah.
But where are you basing that claim? Because I worry in the sort of degradation of even encouraging public discourse, it may actually backlash and say, well, gee, you know, Case Western did a great deliberative democracy approach, but those university people, like, they're not to be trusted, right? So, right? So I'm at Cleveland Clinic, by the way. So I just like your thoughts on that.
No, I think it's a really important issue. I think deliberative democracy, what I like about the notion and the method is that it shows respect for what people really think, but it's not like just, you know, off the top of your head, what do you think? It's designed to get people to really reflect on their values and how this might or might not comport with their values. And in a setting where they're doing it with other people. And they're, you know, the rules of the road are respectfully. There can't be, you know, you respect everybody's view and try to come up with some consensus basically about what should happen. Now, you're absolutely right that it has been done in some places and in small numbers. And it's usually an academic who runs it or puts it on, but it's the people who participate in the deliberative democracy are members of the public. And in the small examples that I'm aware of, it's really been quite valuable. But how to scale it is a real challenge. Yes?
Thank you. That was a wonderful presentation.
Thank you.
And I'd like to say something about the DIY concern,
Okay.
which I think we need to attend to, but it may be fairly modest in comparison to what malevolent societies can implement with far greater resources and far much more funding than I could do in my garage, for example. But I'd also like to ask you a question about where do the rights of a donor, a cell donor, a tissue donor, end at some point after agreeing to donate cells, and things can be done to those cells in an infinite number of ways. At what point, or do they ever cease, and do they become the property of someone else?
That's a great question, and that could be a whole other talk, right? You may or may not be aware that there was several years of debate about revising the federal regulations that govern research funded by the Department of Health and Human Services with respect to this question. Actually more pointed than even your question. The question was, do we have to have consent? Do we have to have people's consent in order to use their specimens or their data? And the current regulations say no. If they're de-identified, in other words, I have a set of data or a set of specimens, I don't know that it's yours and Sharona's and somebody else's, but I can use it for research and use it fruitfully for research. And so the opposite argument of that is, well, wait a minute, in that pile of stuff that you're using is part of me, and so how do we, reconcile that? The way the regulations came down was, de-identifying it takes risks away from the individuals. Other people say you can't really de-identify anything, and so that's kind of a false protection. But they did build into the regulations that they're going to revisit what identifiability means every couple of years or something like that. I do think it's a really even more interesting question when we think about what we use them for, the specimens. So, developing cell lines, developing induced pluripotent stem cells, those have, a kind of power that, a lot of other research with specimens doesn't have. And so I think, that little study that I found, people said, yeah, sure, I think it's valuable research, but you got to tell me that that's what you're going to do with it. And I don't think that's what we're doing at the moment. Yeah.
We're going to have to
Stop.
stop.
Okay, sorry.
Please join us at a reception outside, but first I want to present a plaque to Dr. Grady. You've been a wonderful Schroeder Scholar,
Thank you so much.
and let's thank her.
Very nice.
reception around the corner in the rotunda, and we'd love to see you all there.